Divergent DNA, the accidental discovery that's shaking genetics. Scientists testing a new method of sequencing single cells have unexpectedly changed our understanding of the rules of genetics. The genome of a protist has revealed a seemingly unique divergence in the DNA code signaling the end of a gene, suggesting the need for further research to better understand this group of diverse organisms. The work was intended to test a DNA sequencing pipeline to work with very small amounts of DNA, such as DNA from a single cell. Dr. McGowan was working with a team of scientists at the Earlham Institute and with Professor Thomas Richard's group at the University of Oxford. Unexpected genetic findings in protists. However, when researchers looked at the genetic code, the protist Oligohymenophoria sp. PL0344 turned out to be a novel species with an unlikely change in how its DNA is translated into proteins, Dr. McGowan said. It's sheer luck we chose this protist to test our sequencing pipeline, and it just shows what's out there, highlighting just how little we know about the genetics of protists. It is hard to make any statements about protists as a group. Most are microscopic, single-celled organisms like amoebas, algae, and diatoms, but larger multicellular protists exist such as kelp, slime molds, and red algae. The definition of a protist is loose. Essentially, it is any eukaryotic organism which is not an animal, plant, or fungus, said Dr. McGowan. This is obviously very general, and that's because protists are an extremely variable group. Some are more closely related to animals, some more closely related to plants. There are hunters and prey, parasites and hosts, swimmers, and sitters, and there are those with varied diets while others photosynthesize. Basically, we can make very few generalizations. DNA translation anomalies. DNA is like a blueprint of a building. It does not do anything in and of itself, it provides instructions for work to be done. In order for a gene to have an impact, the blueprint must be read, and then built into a molecule which has a physical effect. For DNA to be read, it is first transcribed into an RNA copy. This copy is taken to another area of the cell where it is translated into amino acids, which are combined to make a three-dimensional molecule. The translation process starts at the DNA start codon, ATG, and finishes at a stop codon, normally TAA, TAG, or TGA. In Oligohymenophoria sp. PL0344, only TGA functions as a stop codon, although Dr. McGowan found there are more TGA codons than expected in the ciliate's DNA, believed to compensate for the loss of the other two. Instead, TAA specifies lysine and TAG specifies glutamic acid. This is extremely unusual, Dr. McGowan said. We're not aware of any other case where these stop codons are linked to two different amino acids. It breaks some of the rules we thought we knew about gene translation. These two codons were thought to be coupled. Scientists attempt to engineer new genetic codes, but they are also out there in nature. There are fascinating things we can find, if we look for them. Or, in this case, when we are not looking for them.